Okay, first of all, I would like to thank the organizers here at USB for putting together this workshop on peptide and oligonucleotide therapeutics. My presentation this morning will be focusing on some of the more recent CMC and regulatory challenges that we have encountered at the FDA in the drug applications for oligonucleotide drugs. But before I start my presentation, I'd like to define a word for you that you'll hear quite frequently in my presentation, and that word is quality. As Mike said earlier, I am a primary reviewer at the FDA in the Office of Pharmaceutical Quality. And for us, a quality product of any kind is something that meets the expectations of the user. And in this case, the user is the patient. For us, we try to help bring about pharmaceutical quality for these new therapeutics such that every dose is safe and effective. This gives assurance to the patient that their next dose will have the same quality as the previous dose. Now, as I mentioned at the FDA, we see or get several different types of drug applications for oligonucleotide therapeutics. The ones that we see currently are the IND, NDA, and BLA. The IND is the first application, the investigational new drug application. This is the application where an investigator or a sponsor request authorization to administer a new therapeutic to humans. This application contains the proposed information for the clinical trials, along with the safety and toxicology information showing that this new medication will be safe once administered to humans. And it also gives us a glance into the quality of the new therapeutic. After an applicant or sponsor has successfully completed their clinical trials, we would see this new therapeutic come in as an NDA, new drug application, or a BLA. The difference between the two is for the NDA, currently for oligos, these are for the oligos that are made through a chemical process, whereas oligos that are made by a vector-based or a promoter-driven process, such as messenger RNA for a vaccine, those come forward as a BLA. For today's presentation, we're just going to talk about some of the issues we see in INDs and NDAs, and we will not be discussing some of the CMC issues for BLAs. Now, the CMC information that we get in the IND and NDA is roughly split between the drug substance, which is the active pharmaceutical ingredient in this new therapeutic, and the drug product, which is the finished dosage that contains the drug substance. For my presentation this morning, we're just going to talk about some of the issues that we have discussions with sponsors and applicants regarding the manufacturing, the characterization, and the control of the drug substance as part of the CMC package for the drug substance. So let's first go to manufacturing. And like the manufacturing of small molecules, for the manufacturing of oligos, we still get a fair amount of questions and have discussions with manufacturers on the designation of the regulatory starting materials. Now, this is important because the designation of the regulatory starting materials signifies the start of the GMP process. At the FDA, we look to the guidance ICH Q11 and its corresponding Q&A for basically the general principles that we use to help designate these regulatory starting materials. Now, fortunately, for the manufacturing of synthetic oligos today, most of them are made chemically on using a solid support. And the starting materials for that solid support synthesis are the phosphoramidates. Manufacturers today have a choice of a very robust supply of the more common phosphoramidates, in which there are substitutions at the 2 position, the 2-hydroxy, the 2-methyl, the 2-fluoro, along with substitutions, of course, at the base as their starting materials for their synthesis. These manufacturers today can provide these materials in high quantity and high quality, such that they come with really all what we're seeking for in terms of the general principles outlined by Q11. And most of all, and most importantly, they come with a defined impurity profile stated in their certificate of analysis. That impurity profile allows us to judge the impact that the impurities in these starting materials will have on the final drug substance. Not all of the oligos come this way. As we get into the more custom synthesized oligos, we're still going to depend heavily upon the specifications for these materials, along with, as I said, that they conform to all the general principles of ICH Q11. This also brings up some of the materials that we're seeing for the solid support. Traditionally, the solid support just has been the support with the linker, and then the manufacturing process, which attaches the phosphoramidates to comprise the oligonucleotide strand. But currently, we're also seeing solid supports that come fully loaded with the first nucleotide as part of the nucleotide sequence, or they may come loaded with a conjugated warhead that will become part of the final drug substance. So in this case, the solid support is actually adding a portion of it to the final drug substance. And so we'll be looking at these solid supports to conform to all the general principles of ICH Q11 in terms of them being a regulatory starting material. In addition, some of the other issues and discussions that we have for manufacturers of oligonucleotides is the use of in-process control data as part of the final drug substance specification. This is especially occurring in the manufacturing of double-stranded oligos, such as siRNA, where we look to have confirmation of the oligos in the oligonucleotide strand. For these double-stranded, it is much easier to confirm the nucleotide sequence not at the final drug substance stage, but earlier when the single strands are manufactured. And while this is permissible under the guidances that we use to regulate oligos, we do ask that the manufacturers do justify that the test results the test results that they're going to use for the in-process control data as part of the drug substance, that they do not change from the in-process stage to the final drug substance. Going on to characterization of the drug substance. Fortunately, the techniques and methods that have been used for years in the characterization of the structure of low molecular weight therapeutics, such as NMR, mass spec, IR, elemental analysis, they can be applied to characterizing and confirming the structure of the oligonucleotide drug substance. However, we still get a fair number of questions on confirming the bioactive conformation of the oligonucleotide. Fortunately, for the single-strand oligonucleotides, such as the antisense class of oligos, their bioactivity is dependent upon their primary structure. In other words, it's dependent upon the nucleotide sequence in the strand. And thus, we look for a confirmation of what that nucleotide sequence is, and that can be done by a mass fragmentation analysis, or by a cap failure sequence analysis, or even a T-melt. But these are some of the special techniques that are needed to confirm the bioactive conformation for single-stranded oligos. For other oligos, such as aptamer, we may need other techniques to confirm the bioactive conformation. For instance, for the double-stranded oligos, circular dichroism data may be more appropriate to confirm that the bioactive conformation can be obtained. For aptamers, you may have to go as far as a bioassay to show that the proper bioactive conformation can be obtained. For impurities, this is more challenging for oligos than it is for low molecular therapeutics. This is mainly because for oligos, due to their large size, the possibility of diastereomers, and also because the physical chemical properties of the final API may be quite close to the physical chemical properties of some of the more common product-related impurities, that today's analytical techniques can't really fully separate and resolve these impurities from the final API. This can be seen quite clearly in these two chromatograms at the lower part of this slide. In the one on the left-hand side, you see quite clearly that there is a main peak for the purified oligonucleotide, in this case, a single strand. And you see a few impurities that have eluted earlier, the early eluting impurities, and maybe a couple that have eluted later, the late eluting impurities that haven't been fully resolved from the main API. But really, there is a lot more substances, product-related impurities that are hiding under that main API peak. And this can be seen in the right-hand chromatogram of a mass spec of that central peak that was on the left-hand side. And there we see a lot of other masses that signify that there are other impurities under the main API peak, that while they cannot be totally removed from the final drug substance, but they can be identified. And manufacturers today have been able to show that this impurity profile, while they can't remove these impurities, they can show that they can be identified and characterized, signifying that their manufacturing process is controlled. And this brings up an interesting point in using a platform data to show that this impurity profile is the same for different for different oligos of the same class, as long as the same manufacturing process is being used. And how to use this platform data. What we're currently in having discussions with amongst ourselves and with various manufacturers is that this platform data is an excellent way to get an early estimate of the acceptance limits for this impurity profile. But there is a lot more other data that goes into the acceptance limits than just the manufacturing capabilities as represented by this chromatogram here. For instance, toxicology data is part of the acceptance limit selection, as well as stability data, safety data, and more importantly, what the planned clinical dose. All those are taken together to choose the appropriate acceptance limits for the impurity profile. But at least platform data is giving you an early start on where those ranges should be. And we look forward to having discussions with manufacturers on how to best use this platform data to get an early estimate on what the acceptance limits should be. Now, I'd like to talk a little bit about the control of the drug substance. And for this, I'd like to really delve into the drug substance specification. Now, the drug substance specification is a set of criteria that are designed to ensure the quality and the consistency from batch to batch for this new drug substance. And we often have discussions with drug substance manufacturers on what are the appropriate guidances that they can use to help them set the tests that are needed along with the proper acceptance criteria. And as I pointed out earlier, the guidances such as ICH Q3A and ICH Q6A, which have been very helpful in putting forward the acceptance criteria and the test for low molecular weight therapeutics. Unfortunately, these guidances don't explicitly cover oligonucleotides. However, we kind of look at these guidances as a starting point for discussions with manufacturers on what the proper test should be in the drug substance specification and what the acceptance criteria should be. Now, some of the other guidances that we have also used in low molecular weight therapeutics, their recommendations, such as the guidance for residual solvents, the guidance for elemental impurities, those do directly apply to oligos. And so the recommendations there can be used directly for oligos. But in terms of the discussions that we have, I want to really drill down and look at the test for identity, assay, and purity. For the identity test in the drug substance specification, this should be a specific test that can discriminate between compounds of closely related structure. This is difficult for oligos because some of the product-related impurities are very close in structure to the final API. So we request for identity for oligos that there be at least 2 identity tests for confirming the identity of the drug substance. For instance, one of the tests can be identity by molecular weight. But we do recommend that one of the identity tests confirm the nucleotide sequence of the drug substance. And that can be done, as I said earlier, by mass spec fragmentation analysis, by cap failure sequence analysis, or by T-melt. But we do recommend that that be part of the identity test in the drug substance. For assay, this is a specific stability indicating test that can determine the content of the new drug substance in the material. Now, for this test, we do want to make sure that to be able to do it properly for oligos, you do need to have a well-defined reference standard of known purity to base the results of your assay test against. Now, for double-stranded oligos, we also recommend that the test be a denaturing test, in which for a denaturing test, that it separates the 2 strands of the double-stranded oligo into their individual strands for a more accurate determination of the content of the new drug substance. And lastly, we still do get a fair number of questions regarding the PO impurity in the drug substance for the oligos that may have a phosphosulfur linkage. And we do look at that substance, that PO substance, as an impurity, not part of the drug substance, even though it may have the same biological activity. but we do look at it as an impurity, and so it should not be included as part of the calculation for the purity in the assay. Lastly, I wanted to talk about the purity test, and we've mentioned a little bit about it in terms of impurities, but the purity test should be a test that quantifies the degree to which other compounds, such as some of the drug-related impurities, are present in the drug substance. And here, for double-stranded oligos like siRNA, we recommend that as part of this test, you use two orthogonal HPLC methods. One would be a denaturing method that separates the final drug substance into its single strands, and also a non-denaturing method. These combined results do give us a better handle on the purity of the final double-stranded oligo, because it shows different impurities that may also still be in the final API. Lastly, in terms of impurities, we did talk about it earlier as part of the characterization, but in the early stages, when we're having discussions with drug substance manufacturers, there's always talk about, are there ways to group the impurities, since sometimes that the resolution between impurities is not high. And so in this case, we'd say, well, one way to at least group the impurities can be either on structural class, such as the short mers or the long mers, or the oligo impurities that have lost one of the nucleobases, or by retention times. This is okay at the early stages at the IND, but as we get to the final NDA, we want to see a lot more of the identification of the impurities, such that showing that your manufacturing process is controlled. And lastly, it bears repeating because we do not have the same types of guidances for oligos that we have for low molecular weight therapeutics. But at this time, the thresholds that have been used for low molecular weight therapeutics, such as for reporting, identification, and qualification, those thresholds have not been set by the FDA. So we're still kind of we prefer to have look at this as a case-by-case basis and have discussions with manufacturers on what kind of the types of data that they are using to set the acceptance limits for the impurities in their drug substance. So in closing, we have talked about some of the more common areas that we have discussions with manufacturers of synthetic oleos, that being the issues around manufacturing, characterization, and the drug substance specification. And we have talked that not all the guidances that we have used in the past for low molecular weight therapeutics, while they do not specifically cover oligos, they are a good starting point for discussions and guidelines on where some of these limits should be put. And like I said, it's still on a case-by-case basis, and we do look forward to having discussions with manufacturers on getting to acceptable limits to ensure the quality of the final drug substance. And lastly, I would like to say that we are working on new guidances for oligos. Hopefully, coming out in 2021 is guidances for investigators who are treating only a small group of patients. Sometimes we call these N=1, but patients who have a debilitating genetic disease. And in this case, we are trying to outline for these investigators what are some of the CMC information they need so that they can rapidly get this medication to treat their 1 or 2 patients.